Hi everyone. Today we will be discussing the ethical and biosafety issue related to invasive genetic sampling in the critically endangered howling crow or alala. This bird, once extinct in the wild, is now the focus of intensive conservation effort. Because of its small population and low genetic diversity, monitoring its gene is vital for guiding breeding and reintroduction. However, many genetic sampling techniques are invasive, requiring blood or tissue, which can stress or harm the animals. In population where every individual matters, even minor injuries or stress can have serious consequences. This raises serious bioethical questions about how we balance conservation science with animal welfare. In the following slide, we will explore how the principle of 3R, replacement, reduction, and refinement, can offer solutions to reduce the impact of genetic study on this endangered species.
Firstly, we will start with the replacement method for this chicken surrogate system for the Alola species. The methods includes development of stage method by using the CRISPR-Cas9 components transfected into the sperm, where the edited sperm will fertilize the eggs and produce the gene-edited chick in one generation. The ethical benefit of this replacement method includes the direct gene editing trials on endangered species with reduced risk of embryo loss or unintended mutation in rare animals. The limitations include the genetic deletions might be observed on certain private sites, and the efficiency of gene editing was low, with the potential of target effects and variable transmission of edits from founders to progeny. So in short, chickens share similar development and genetic traits with other birds. However, with this replacement method, this allows researchers to test gene editing precision, efficiency, and safety without harming endangered species like the alala.
Next, for reduction, is the in silico simulation of gene drives. Alright, so what is in silico simulation of gene drives? Basically, it's using computer models to predict how a genetically engineered trait, like one that spreads disease resistance, would move through a population without actually releasing anything into the wild. How do we do it? Scientists use tools like individual-based models and stochastic simulation to plot in real biological data like mating rates, mutation changes, and movement patterns, and run scenarios into see what might happen over time. Why does it work? Because it's safe, cheap, and smart. Ethically, it's a win. No animals harmed, no habitat messed up. It gives us a way to make informed decisions without taking reckless steps.
For the refinement method, we will use the high-fidelity CRISPR-Cas9 variants, and for this method, we utilize the modified enzymes such as HSP-Cas9-HF1 and ESP-Cas9 to improve the DNA binding accuracy. These versions of enzymes will introduce amino acid mutations in Cas9 to reduce the binding of target sites, and it will still recognize the correct DNA sequence. For the ethical benefits, it minimizes the genetic harm in small or sensitive population with the refinement of editing process for greater safety. The limitation of these methods include the slightly decrease on target editing efficiency, and it can fail to efficiently edit certain genomic sites with complex chromatin structures. So in short, wild-type Cas9 can cut similar but incorrect sites, causing harmful mutations, and the high-fidelity variants can retain genetic editing activity but reduce off-target mutations by up to 24.
For the alternative is the comparative transcriptomics and immune profiling. So comparative transcriptomics is a powerful non-invasive method that allows researchers to analyze and compare gene expression pattern across different species or conditions. In this case of the alala, it involves examining RNA sequences from disease-resistant and susceptible birds to identify key immune-related genes without altering their DNA in any way. The process starts with RNA collection from relevant tissues like blood or spleen, followed by RNA sequencing using high-throughput technologies. Researchers then analyze the expression data to identify differentially expressed genes and map them to known immune systems. This approach is highly effective because it provides deep insight into immune function and disease resistance while avoiding the risk of genetic modification. From an ethical standpoint, it aligns beautifully with the 3Rs: replacement, no need for genetic manipulation; reduction, maximize data per sample; and refinement, minimally invasive, low-stress method, promote animal welfare.
Invasive genetic sampling poses significant ethical and biosafety concerns for the critically endangered alala, a species already on the brink of extinction. By embracing the principle of the 3R, replacement, reduction, and refinement, we can shift to a more human, sustainable, and scientifically advanced method. This approach not only protects individual birds from harm and stress, but also enhances the quality of conservation genetic research, ensuring the long-term survival of the alala without compromising its welfare. That's all from our group. Thank you.